Good morning, friends. Today we will be studying about physiology of urine formation, which is a topic of Masters of Zoology semester 2 of Ranchi University syllabus. Urine is basically an aqueous solution of nitrogenous wastes and comprises of various inorganic salts such as sodium, potassium, calcium, and magnesium, which are present in various forms. The peculiar color of urine is due to the presence of urochrome, which is a breakdown product of hemoglobin. And although the only source of urine is blood plasma, but the composition of both plasma and urine are completely different. Urine formation basically involves three steps of glomerular filtration, reabsorption, and tubular secretion. Now, as we know that urine formation takes place in kidney, and the basic structure and functional unit of kidney is nephron. The diagram shows the structure of nephron, and the uppermost structure is Bowman's capsule, which comprises of capillary bed, that is the network of blood capillaries comprising of afferent arteriole and efferent arteriole. From Bowman's capsule, we move to the proximal convoluted tubule, also known as PCT, and it comprises of two limbs, descending limb and ascending limb, and between both the limbs is present the loop of Henle. Then we come to distal convoluted tubule, also known as DCT, and finally to the collecting duct. So the first process is glomerular filtration that takes place in the capillary network. Now, as you can see in the diagram, this is the fine structure of glomerular capillary. The first layer is endothelium, also known as lamina fenestrae. Then comes the glomerular basement membrane. Beneath the basement membrane is podocyte. These are certain specialized cells that are present in the visceral layer of Bowman's capsule. Podocytes comprise of numerous food processes known as pedicles, and there are space present between these pedicles which are very narrow, of about 100 angstrom, and are known as slit pores, where the actual ultrafiltration of plasma proteins takes place. And beneath the podocytes is the outer lining of Bowman's capsule, and between the podocytes and Bowman's capsule lies the lumen of Bowman's capsule. Although the capillary endothelium does not take part in ultrafiltration, but it exposes the plasma to basement membrane. Ultrafiltration is basically a passive process and involves various kind of pressures that determine the ultrafiltration process. The diagram shows various pressures that are involved and are present in the glomerulus area. First is the hydrostatic pressure, that is 55 mm of Hg. Then is blood colloidal osmotic pressure, 30 mm of Hg. Then is effective filtration of pressure, that is 25 mm of Hg. And there is renal intratubular pressure, which is 10 mm of Hg, and intracapsular hydrostatic pressure of 15 mm Hg. Next, we come to autoregulation of glomerular filtration. There are basic two mechanisms. First is myogenic mechanisms. In this case, when there is increase in blood pressure, there is stretching of afferent arterioles, which increase the blood flow into the glomerulus. And when the blood has entered the glomerulus, the afferent arteriole again undergo contraction, and thereby reducing the blood flow. So the complete regulation of blood flow takes place by myogenic mechanism. Second is juxtaglomerular apparatus, JGA, which is a group of cells located between DCT, that is distal convoluted tubule, and afferent arterioles. These cells secrete enzymes known as renin, and renin are responsible for modulating blood pressure, thereby regulating the blood flow into the glomerulus. Now, the composition of glomerulus filtrate is glucose, amino acids, vitamins, water, mineral salts, hormones, urea, and creatine. And after filtration, the substances that has been removed from the filtrates are RBC, WBC, blood platelets, and plasma proteins. Next process is selective reabsorption. Now, after filtration, almost 170 liters of solution per day is being filtered in a normal man, but the total amount of urine excreted out of the body is only 1.5 liter. So, rest of about 99% of glomerulus filtrate is being reabsorbed, and this process is known as selective reabsorption. About 80 to 87% is reabsorbed in the proximal convoluted tubule by the process of obligatory reabsorption, whereas 13 to 20% is reabsorbed by distal convoluted tubule by facultative reabsorption. Coming to the role of proximal convoluted tubule in selective reabsorption, about 80% of glomerular filtrate is being reabsorbed in the PCT, and the substances that are reabsorbed are basically total amount of glucose, most amino acids, all vitamins, all hormones, 70% of sodium ions, 75% of potassium ions, large number of calcium ions, 75% of chloride ions, 80% of water, and 50% of urea, out of which chlorine, water, and urea are being reabsorbed by passive transport. Next, we come to role of loop of Henle in selective reabsorption. Now, the basic function of loop of Henle is to concentrate the urine and make it hypotonic than blood. It basically increases the sodium chloride concentration in medullary region so as to extract water from collecting tubule. Next is reabsorption of glucose, like how glucose is being reabsorbed in the nephron. It is completely reabsorbed in the proximal convoluted tubule, and the process is against concentration gradient. Its reabsorption is carrier mediated, that is, it depends on the concentration of carrier proteins that are present in the cytoplasm of tubular cells, and the reabsorption also depends upon the concentration of sodium ions in the tubules. Next is reabsorption of proteins. Now, although the protein is filtered during glomerulus filtration, but a small amount is being reached to the convoluted tubule, which is reabsorbed by the process of pinocytosis. The plasma membrane engulfs proteins and forms pinocytotic vesicles, and these are digested by lysosomal enzymes. In selective reabsorption process, we will also have to study the counter current theory. According to Nelson 1958 and Regal 1972, the ability of mammalian kidney to form concentrated urine is due to the hairpin-like structure of loop of Henle, which comprises of descending limb and ascending limb. Both these limbs form a hairpin countercurrent system because the fluid in them flows in opposite direction. First, we will be seeing about the descending limb. Descending limb is thin and freely permeable to water, but it is not permeable to salt, that is sodium chloride. Sodium chloride is concentrated in filtrate due to osmotic loss of water from the filtrate as the descending limb moves down to the renal medullary region. The filtrate that enters from PCT to descending limb is isotonic to blood, but when this filtrate passes through the descending limb, 5% of water flows out due to osmosis and is reabsorbed by the vasodictor. Now the filtrate becomes hypotonic in nature, and thus the filtrate becomes more and more concentrated. Ascending limb, on the other hand, is completely opposite to that of descending limb. It is, although a thin segment, but is impermeable to water and is permeable to sodium chloride. As the filtrate enters the ascending limb, sodium chloride is actively removed to the medullary region, from where it enters passively into the descending limb. Water does not move out, as the ascending limb is impermeable to water. We discussed this. The concentration of salt is maximum to the region where collecting ducts open into renal pelvis in the deeper medullary region. The higher concentration of salt in medulla results in outer movement of water from collecting duct to medulla, making the urine more concentrated. Next is the role of distal convoluted tubule and collecting tubule. In these regions, sodium and water are reabsorbed. Sodium is reabsorbed actively and potassium is excreted in the urine. Chloride ions are also reabsorbed but by passive process. Role of collecting duct. Now, because collecting duct is lined by cuboidal epithelium, it is permeable to water but not to sodium chloride, and it takes the filtrate to the medullary region for the second time, and the filtrate becomes more and more concentrated. In the diagram, it can be seen that the proximal convoluted tubule uh, reabsorbs 80% of filtrate. The descending limb is permeable to H2O and NaCl both, and the filtrate is hypertonic. In case of ascending limb, it is impermeable to both water and NaCl, and the filtrate is hypotonic in nature. Whereas in case of DCT, reabsorption of sodium takes place, whereas excretion of potassium takes place. Collecting duct leads to diffusion of water, and the urine obtained is hypotonic in nature. The third process of urine formation is tubular secretion. Most of the tubular secretion takes place in the distal convoluted tubule, that is the DCT, and the selective ions such as potassium, sodium, and hydrogen ions are, and some drugs and metabolic waste such as creatinine and ammonia are actively transported into the tubule. Most of the potassium is secreted into DCT in exchange of reabsorption of sodium, and uric acid and ammonia are also secreted in distal convoluted tubule and collecting tubule. So that is all for physiology of urine formation. Thank you.